and have this process in its base. It lines the ventricles of the brain. This is ventricles present in our brain. A mesenchymal cell line these ventricles. They assist in the formation of the cerebrospinal fluid. They assist in the formation of the cerebrospinal fluid. Satellite cell. This is one of the supportive cells present in the peripheral nervous system. They form thin layer around what? Around what? Around the cells present, nerve cell present in the ganglia, like sympathetic ganglia or sympathetic or spinal ganglia. What is the function of the satellite cell? Protection, insulation, and nutrition. Okay, now we will discuss degeneration and regeneration of the nervous tissue. Damage to the nerves in the central nervous system is permanent. There is no regeneration. Injury to the peripheral nerves is followed by regeneration. This occurs in the peripheral nervous system. Degeneration and regeneration how occur. Now we will discuss degeneration. Cut of a peripheral nerve will include traumatic injury at the site of the injury. Retrograde degeneration, it occurs retrograde in the cell body. Wallerian degeneration distal to the site of injury. So this part called traumatic degeneration at the site. This called retrograde degeneration in the cell body, called retrograde degeneration. Wallerian degeneration occur in this part in the distal segment. Retrograde degeneration mean changes which occur in the cell body after injury of its axon. These changes include what? Include cell will be swelling in the cell, withdrawal of its process, there is no process, and become spherical. The cell become spherical. Nucleus become eccentric and pycnotic. Nasil granules, the nasil granule break down into fine granule, then disappear. What's called chromatolysis. What is mean by chromatolysis? Nasil granule disappear and break down into fine granule, and finally it disappear. Golgi apparatus also disintegrate and finally disappear. Neurofibrils disintegrate and finally disappear. Finally, the cell body degenerate and its axon undergo what's called Wallerian degeneration. What is mean by Wallerian degeneration? It is the changes that occur in the distal segment. Include what? The axon undergo rapid degeneration in few days. Myelin sheath also fragmented into what? Into fat droplets. So the degeneration occur at the axon and the myelin sheath. Schwann cell or neurolemmal sheath don't degenerate. It's very important to know that Schwann cell don't degenerate. It proliferate to form thick tube. Schwann cell, it will form thick tube. This tube will act as guide for the regeneration to aid the regenerating axon. This tube formed by the Schwann cell, it acts as guide for the regenerating axon. The axon will sprout from the proximal stump of the cut nerve. Regenerations. What is the factor affecting the regeneration? The presence of the neurolemmal sheath, which means the presence of the Schwann cell. The nerve fiber must be small cut, distance between them. The nerve fiber is partially cut, not severely cut. Mechanisms of regeneration, which at first neurolemmal sheath or Schwann cell proliferate. At first, the nerve fiber will reform its normal structure. Several branches grow at a rate of 1 mil per day. Several branches from the axon will grow at a rate of 1 mil per day. Several axons enter the tube. Only the proper tube will reach the nerve ending. Schwann cell at last will form the myelin sheath. Now, one of the following share in the formation of the blood-brain barrier. What? Which cell? astrocyte. One of the following is important in nerve regeneration. Which cell is very important? Is Schwann cell. Give reason for degeneration of the central nervous system is permanent. Why? Why there is permanent? Because there is no Schwann cell. There is no Schwann cell in the central nervous system is important, as the Schwann cell is very important for nerve regeneration.